children. Welcome to the online class of biology. So, we were studying about various topics like transcription, replication, etc. Okay, so today I'm going to explain you about the genetic code, okay, and its discovery, and also we will study about the tRNA molecule. Okay, so you have already seen that the RNA synthesis is taking place according to the complementarity, isn't it? That is, the information that is there in the DNA, okay, RNA will be synthesized based on the complementarity. That is, if where A is there in the RNA molecule, U will be added, and if T is there in the RNA molecule, a new nucleotide of the means A will be added. If T is there, A will be added. If A is there, then instead of thymine, uracil will be added. In this way, what you can see that complementarity exists between DNA and RNA. But in case of protein synthesis, so protein synthesis is called what? Translation. That is the synthesis of proteins from RNA. By reading the information from RNA is called what? The translation. And there is no existence of any complementarity between RNA molecule and the proteins or amino acid sequences. And at the same time, okay, there if any change happens in the RNA molecule or DNA molecule, that will affect the sequences of amino acids. Okay, so this finding resulted in more experiments, and that resulted in the finding of genetic code. Okay, so for this, from several disciplines like physics, organic chemistry, okay, biochemistry, genetics, so from all this, so many scientists, okay, they have contributed their means they were also working on it. And later on, a scientist known as George Gamow, a physicist, who only said that if there are 4 bases, okay, for 20 amino acids, 20 amino acids are there, and if there are only 4 codes, 4 codes means what? Adenine, guanine, okay, then what? Uracil, okay, and what? Cytidine. Okay, so like this, cytidine, like this, if 4 codes are only there, and if these 4 codes has to code for 20 amino acid, there should be combination of bases. Okay, the code should have combination of bases. Okay, so he proposed that, okay, that is, the code should have three nucleotides. Okay, is it clear? That's what he suggested. So what you understood is, like 26 alphabets of English can make a number of words, isn't it? So in the same way, with different combinations, this 4 bases should be able to code for this 20 amino acids. Understood? So like if the codon or was having only 2, then it is not possible to code for all the 20 amino acids. So if it is 3 letters, then the permutation combination can give, okay, 64 codons, and this 64 is enough for coding 20 amino acids. Understood? The combination of means the combination of 3 nucleotide sequences can easily code for 20 amino acids, as it is allowing 20 amino acids to be coded. Understood? So genetic code means what? It is the nucleotide sequences in the mRNA that codes for a particular amino acid. Is it clear? Means genetic code means all the codons. Okay, sequence of codons. And a codon contains how many letters? 3 letters. Okay, or 3 nucleotides like ATC or GCT. Understood? So in this way, 3 letters will determine one amino acid. Okay, the codes in the mRNA which codes for, or the triplet codons which code for one amino acid. Understood? I told DNA information in the DNA, first it will be transcribed to RNA. I told DNA cannot directly come out from the nucleus and direct the synthesis of proteins. So it has to do what? It has to make a RNA molecule, and in the RNA molecule also what will be there? Nucleotide sequences will be there. So in this, 3 letters will code for one amino acid. Understood? So during translation is what? The synthesis of proteins or polypeptide chain in where it takes place? In the ribosomes, after reading the information of the RNA. So in the RNA, how the information is stored? In the form of codons. Okay, so codons, I told, it contains 3 letters. 3-letter codons they are. Okay, who proposed it? It is proposed by George Camus. Okay, so he said that there should be, if only 4 bases are there, and if that 4 bases, what are the 4 bases? A, U, C, G. Isn't it? So if they have to code for all the 20 amino acids, there should be combination of this. So if only 4 bases, if they are single, okay, means A if it is code for one amino acid, and U if it is code for another amino acid. Likewise, if it is, then only 4 amino acids can be coded, isn't it? So he suggested that it should be a combination, and if it is 2 also, it is not sufficient. Okay, so he suggested that the the codons should be a combination of nucleotides, and it should be 3. Okay, so if it is 3, the permutation combination, that is 4 raised to the power 3, that is 4 into 4 into 4 is equal to 64 codons, and that is enough, well enough for coding 20 amino acids. Okay, so George Camus explained the code should have combination of 3 bases. Okay, and the experimental proof was given by Hargobind Khurana. Okay, Hargobind Khurana has given the proof? By synthesizing RNA molecules with defined combination of bases. Okay, so Hargobind Khurana, he was an Indian scientist. Okay, so he could give evidence by synthesizing RNA molecule. Okay, then another scientist known as Marshall Nierenberg, he could synthesize protein in a cell-free system, means without outside the cell he could synthesize protein or amino acid chains. Okay, and in this process an enzyme was used, that is Severo Okao enzyme. Okay, so Okoa enzyme, they use polynucleotide phosphorylase enzyme. Okay, so this was helpful in polymerizing RNA. So this scientist, okay, they synthesized RNA molecule, understood, by the help of this enzyme, and then in a cell-free medium. Okay, when they had given this RNA molecule, they could see that what all amino acid chains are or amino acids are added. So from this they could decipher the genetic code. Understood? So when you see the genetic code, you can see this is the genetic code, okay, of existing in the genes, okay, or in the genetic system. You can see that here UUU, if in an mRNA, if UUU is there, then the amino acid coded will be phenylalanine. Okay, and UUC, you can see phenylalanine will be formed. UUA, then leucine. You see, instead of U or C, if it comes an A, then another amino acid will be coded. I told now. So if any small changes happens, in this way only mutation takes place. Instead of phenylalanine, what will be added then? Leucine. UUG, if it is, then also it is leucine. Understood? Then you see here like this, you can just see all this, then you can understand what all amino acids. So in mRNA, if the amino acids, the nucleotide or genetic code is like this, UUG, then leucine will be there. And if it is CCA, okay, CCA or CAC, then histine, okay, histone will be produced, okay. Then what, GGA, UGC, then cysteine. And you can see UGA, it is a stop codon, okay, it will be a stop codon. And you see other likewise stop codons are there, UAA, UAG and UGA, these are stop codons. And AUG, you can see AUG will be producing a amino acid, or it will be coding an amino acid called methionine, and it also acts as a starting codon, okay, initiator. Understood? So these are what different the codons or genetic code. Okay, so this is the genetic code, means in the mRNA, if this triplet codes comes in the amino acid or in the polypeptide chain, the amino acid added also will be specific. So if you see the salient features of genetic code, the genetic code is triplet. Okay, triplet means it is having a genetic code or codon will have 3 letters. Okay, and then 61 codons can code for amino acids, and 3 codons they will not function or they will not code for any amino acids, and they are known as stop codons. Okay, and some amino acids can be coded by more than one codon. That is what it is. The genetic code is said to be degenerate. Okay, what is degenerate means? Some amino acids can be, for example, you see here leucine. Okay, same amino acid can be what coded by a number of codons, CUU, CUC, CUA and CUG. So this property of genetic code is known as what? Degenerate. Okay, genetic code is degenerate, and this is actually helpful. How it is helpful? Because if a small single nucleotide mutation happens, then also there won't be any change in the amino acid. Okay, so it's a what additional advantage. Then the codon is read in mRNA in a continuous fashion. Contiguous fashion. Okay, contiguous or continuous fashion you can say, without any comma or punctuation. So any mutation happens, then it will be problematic, isn't it? So that will be the next letter will be taken as the 3 letters. Okay, without any punctuation or full stop, it will be just read continuously. Then the code is universal, that is from bacteria to human being or from any organism you take. Okay, the codons will be similar. Okay, that is UUU will code for phenylalanine only. Understood? This is one example only, but some exceptions are there, found out later in mitochondrial codons. Mitochondria is also having DNA, and in that and also in some protozoans. Clear? AUG is having dual function. Dual means what? 2. So what are the 2 functions? That is AUG helps in coding for an amino acid called methionine, and also it acts as the starting codon or initiator codon. And UAA, UAG, UGA, these are known as the stop codons or terminator codons. Okay, you have to learn all this. This characters you have to learn every time. This questions based on this are found. Understood? So it is very important to study the characteristics of codons, genetic codons. So now you would have understood how mutations occurs, isn't it? So when we have learned about sickle cell anemia, that time we have learned that a single base pair change will lead to the replacement of amino acid, isn't it? So what happens? Change of amino acid residue glutamine to valine causes what? Sickle cell anemia, isn't it? A single code or nucleotide, if it is changed, then that results in the change in amino acid sequence, and that will change the property of protein, and that can lead to severe effects in the body. Understood? So like you can just take the example here, that is here Ram has red cap. Okay, so the same sentence, if one B is added there, what will happen? I told without any punctuation it will be read continuously. So you can see Ram has, then B, if it is come BRE, will be taken as the codon, and you see the entire meaning of or the amino acid, if it is in the DNA or mRNA, the entire sequence of amino acid also will get changed, isn't it? So like this, if one or two nucleotides, okay, sometimes 2 letters can be added, sometimes 3 letters. So sometimes if it is lost also, okay, if one or nucleotides, if it is lost also, the same problem can happen. Understood? Because of what? Continuous reading without any punctuation. Okay, so insertion or deletions of one or two bases changes the reading frame from the point of insertion or deletion. Understood? So that kind of mutations are known as what? Frame shift insertion or deletion mutations. Okay, so insertion or deletion of a particular nucleotide will just change all the sequences from that point, from where they are added or from where they are deleted. From there, the entire sequence will get changed, and this is known as frame shift insertion or frame shift deletion mutations. Okay, so this can cause what? Mutations. Now it is very clear how it is forming, isn't it? How this insertion or deletion causes what? Mutation in the cells. Okay, mutation in the organism. Is it clear to everyone? So as we have studied, okay, DNA, the information from DNA has to be copied finally into proteins, isn't it? Amino acid sequences in the proteins. But DNA is not directly what controlling the process. So Crick itself, Watson and Crick, okay, that Crick was clear that there should be some mechanism to read the code and also to link it to the amino acids. Okay, so he postulated that there should be some adapter molecule. So what do you mean by adapter molecule? That can read the information on the DNA, and at the same time it should be able to give that information to the amino acid. Okay, so adapter molecule will on one hand it will read the code, and on the other hand would bind to specific amino acid. Okay, and it was found out later that this role is done by tRNA. Okay, so tRNA was known before also, before the genetic code was postulated, but its role, how it is useful in this process was not known. So when you see the structure of a tRNA, okay, it has an anticodon loop that binds on the cod. Cod is where? In the mRNA. Understood? So there should be an anticodon site, okay, that can be complementary to the codons. For example, this is the codon AGU, isn't it? So the anticodon here will be UCA. UCA. Understood? So there should be an anticodon loop that will have complementary to the code, and it should have an amino acid acceptor site. For example, here, okay, amino acid should be joined to the other site. Okay, then each of this should have they are very specific. Okay, if 20 amino acids are there will be 20 different tRNA. Understood? And there will be an initiated tRNA. Okay, initiated tRNA which codes for what? Methionine. Understood? So like this, there are different kinds of tRNA. So what we have learned here is structure of tRNA. So this structure looks like a clover leaf. Okay, the structure, the most accepted structure of tRNA is cloverleaf model. In that, it looks like a inverted L. Okay, inverted L. So this also can be asked. This also you have to practice. Okay, so in a tRNA, there should be a site that can complementarily base pair with the codons. Okay, that is known as anticodon. So the nucleotide sequences present in tRNA is called what? Anticodon. That will be complementary to what? Codons. Okay, and it should have a amino acid accepting site also. Understood? So this are what the structure of tRNA. So we will have to study translation in the next class. So what we have studied today is the genetic code. Okay, the genetic code, George Gamow explained the code should have combination of 3 bases. Then Hargobind Khorana, he was experimental in synthesizing RNA. Marshall Nierenberg's, okay, he could produce protein in a cell-free medium. And for this experiment, Severo-Ochoa enzyme was useful. Then we have studied the characteristics of codons or genetic code. They will be triplet, unambiguous, okay, degenerate, no punctuations, etc. Isn't it? And universal, and AUG is the initiator codons, and there are 3 stop codons. And we have also studied about frame shift mutations, and I have also explained you tRNA as the adapter molecule and its structure. Okay, why tRNA is known as the adapter molecule and the structure of tRNA. This much we have studied. Okay, we'll continue in the next class. Thank you very much.